Hello, good evening, everyone. I'm Tanya from Dr. Omics. So, in today's session, I'm going to start with the new module that is a DNA-seq data analysis. So, before we move with this module, can you please little bit tell me about yourself, which background you guys are having?
Hi, I am Abhishek Konar,
Yeah.
and now I am a research scholar in biotechnology. I am from West Bengal, India.
Okay.
Thank you, ma'am.
Akansha, what about you?
I'm a PhD student in Bharati Vidyapit, Pune.
Okay. Mamta ma'am.
Yeah, hi. Good evening, everyone. My name is Dr. Mamta. I'm working as a faculty in Department of Biotechnology. I'm from Mysore, Karnataka.
Saima ma'am. Hi, Rakhi. What about you?
Yes, ma'am. We did class before.
Yeah, yeah.
I did masters in biotechnology.
Hello, hi ma'am. Myself Sam. I'm working in a lab in NGS department in isolation section here in lab.
Okay.
Okay, ma'am.
Can you see my screen?
Yes, ma'am.
So, DNA-seq, that also we call it as the variant calling analysis. So, before we proceed further, can you please tell me anything regarding this DNA-seq data analysis? Anything you know about this? Saima ma'am, are you like you told me like you're working in NGS department? Might be you like guys are handling this type of data, right?
Ma'am, I'm working in NGS department in isolation section.
Okay, in the isolation. Okay. What about others? Any idea about this DNSSEC? See, the main motto of this DNSSEC is that we are going to find out the mutation
हाँ
that is
जावेद
why we are calling it as the variant calling. Okay. And
नहीं नहीं
also
उधर नहीं
you
जा
can
रही नहीं नहीं उधर नहीं जा रही
please mute yourself, guys. So here we are going to discuss about the mutation detection. Might be it is of SNP, that is the single nucleotide polymorphism, or indel calling, you can also call it as, that is the insertion and the deletion. Okay, so before we talk about this DNA-seq, so what is sequencing? We will start with the basic part first. So what is the sequencing, guys? What we can do with the help of the sequencing?
Ma'am, arrangement.
Understand the nucleotide sequences
Yeah.
present in a stretch of DNA.
Yes, right. Sequencing is to figure out the order of DNA and the RNA nucleotide. That means how the nucleotide is arranged in DNA or the RNA. The pattern we want to know that we can understand with the help of the sequencing, right? And also, with the help of the sequencing, we can also understand about the genetic information. So large amount of information which is obtained from the sequencing has deepened our understanding and the fundamental knowledge of the organism, because if you know the particular sequence, then only you will able to get the information of that particular organism. So let's consider person is suffering from a disease or maybe any cancer. So cancer is happening due to some mutation or the variation in his body, right? And you want to find out at which particular position the nucleotide is getting changed, right? And that particular change of the nucleotide, it is causing what? The variation in the DNA. So first of all, you need to know the sequence of that person, and once you know the disease person sequence, then what you will do? You will compare that particular sequence with the reference genome of the human. Then you can understand at which position there is a variation, right? For example, this is a sample of a disease one. We are isolating a DNA, right? Then what we are doing with the help of the sequencing or the alignment pattern, we are arranging this particular DNA on the reference genome. So this sample has been taken from human, right? See, we already know there is a reference genome of the human. So this particular disease sequence, we are going to align on this reference genome. Okay. So for example, on this particular position, there is A, that is the adenine, right? So on this first position, when we are aligning this particular disease DNA on this reference genome, what we saw on this particular position on this disease, it is having the G. So you can see when we are comparing the disease person with the normal sequence, we can see the mutation, right? A is getting replaced with the G, guanine, right? So that we can say this is what? The mutation in the DNA, right? So that we are going to see here in the DNA seq. And with this help, this knowledge will also help the doctor to customize the treatment for the patient. How? Like there are two patients and both are suffering from cancer, and they both are getting some same medication. One person is responding to that medication, but another person is not responding. Okay, so you need to find out what is the change in the DNA of that person, and you will get the idea by looking at that sequence where there is a change of base. So this is how it is helpful for the doctor to customize their treatment. So this is why the sequencing is important, and this is how it is evolved. Okay, so there are three generation of sequencing, and that is how the sequencing has evolved. So let's talk about the first generation. So any idea regarding this first generation sequencing?
It is Sanger sequencing, right?
Who's this?
Akanksha.
Yeah, right. So first generation, we call it as Sanger sequencing. which is developed in the 1975. So it is one of the traditional method, or you can say it is the wet lab technique. So this first generation sequencing, they are using some ddNTPs, which is a dideoxynucleotide triphosphate, which is one of the radioactive isotope label, which is followed by the electrophoresis technique, and where you can detect your sample and also the order of the nucleotide. But the disadvantage, like Akansha ma'am, can you tell me what is the disadvantage of this first generation?
It is applicable for shorter stretches only because
Hmm.
it is a wet lab technique and cannot sequence longer stretches of DNA.
Yeah. So disadvantage, as you said, like it is a low throughput technique. What is that? High throughput or low throughput? Any idea? So in scientific term, we are calling it as the low throughput. As Akansha ma'am said, like it is able to sequence only a stretch of sequence of the DNA, right? So that is why it is going to read only the small reads. That is the first disadvantage of using this first generation, and it require high cost chemical. So some people cannot afford because they are having some budget of their research, right? So that is why like this is the main disadvantage, because it is causing high. And for example, now you have 1,000 of genes, so it is not possible to sequence that 1,000 of genes by this Sanger technique at one time. That is why the accuracy of this technique is low. Okay, that is why people move to the second generation. So now what is the second generation? Kancha ma'am, what is a second generation? What about others? Do you have any idea? Saima ma'am, Sarvanan sir, any idea regarding the second generation? So we call it as the NGS. So have you ever heard about this next generation sequencing? So it contains some sequencing platform, or you can say the sequencing machine like Illumina, Roche 454, ABI Solid, Solexa, Iron Torrent. So with this machines, we are performing what the sequencing in the next generation sequencing. So each machine, it is having its own error rate, and it is only able to read the short sequence. So it is not able to read a complete sequence. So for example, you are having a like a genome of a human, okay, and we want to study the sequence of that particular genome. So what happened after isolation of this particular DNA? What we are doing next step, we are doing the fragmentation. Okay, then after having the fragmentation, we are ligating the adapters, then we are performing the sequencing. So why we are creating the fragments? Because none of the sequencing machine is not able to read this entire genome till yet. Okay, so this is the disadvantage of this NGS. And also in the NGS, it is having the low cost, and there is a high speed of sequencing. And it is causing high accuracy is maintained. There is a high capacity with the sample. And it is able to sequence thousands of genes at a time. So that is why this NGS, we call it as the high throughput technique. That is why like people have like started using this NGS right now. So this is one of the traditional method, and nowadays people have moved to this high throughput technique. So got it why people have moved to this NGS? Clear what is low throughput and high throughput technique? Did you got the difference?
Yes.
Okay. Next is the third generation. Actually, it contain a sequencing platform like PacBio, Nanopore, or there are like many other advanced machine which we are using here in this third generation sequencing. So it is the advanced machine, and it is having a capability to produce long reads. It can be more than 1000 base pair in length, and it is having a high error rate. So any idea why it is having a high error rate? Any idea? So this third generation sequence, what it is doing, it is reading the complete genome without doing the fragmentation step. Okay, and still they are not successful in this like till now. And you can only study the high eukaryotes in the third generation sequencing. And nowadays you can also study the bacterial virus genome also here. But still people are not relying on this third generation sequencing. They are still undergoing in the active development. Okay, so did you got it why it is causing the higher rate? Got it? This three generation. Any doubt till here? Any one of you? So now we will be focusing of the like on NGS, and this is divided into three main steps. So any idea like the three important step in the NGS? Any idea? So first step is the library
Ma'am,
preparation.
hello, I'm audible. Ma'am, library preparation, DNA sequencing, and the data analysis are the three steps in the NGS that we perform.
Yes, right.
Actually, ma'am, I'm outside. I've just leave from the office on
Okay,
the way.
okay.
That's why I'm not able to respond to your questions.
No problem.
Um,
No problem, ma'am.
yes, ma'am.
Okay, fine. Yeah, so we will discuss now. For example, again, there is a cell sample here. And from this sample, like we are going to isolate what? The DNA, right? So this is the DNA. And now, like I already told you, like there is no sequencing platform which can read the entire DNA. So that is why the next step is what? The fragmentations. So this is about the DNA. What if like you have isolate the RNA, right? So once you have isolate the RNA from the sample, we are converting this to the cDNA. Why we are converting this RNA to cDNA? Any idea? With the help of reverse transcriptase enzyme.
To achieve more stabilizing
Yeah.
state, or else it will keep on change its bond breaking will happen. To avoid this, they will convert to cDNA followed by microchip kind of.
Yeah. So see, you already know this RNA. If you are providing a high temperature, it can degrade easily, and also it is unstable than the DNA, right? So that is why we are converting this RNA to the cDNA with the help of reverse transcriptase enzyme. So again, it cannot read the entire DNA. So what we are doing, we are making the fragments here. So once you have the fragmentation, so this is what the fragmentation step. Then we are doing what? The adapter ligation. So we are attaching what? The adapters. So what is the adapters?
Smaller stretches of DNA whose sequence is known previously.
Yeah. So these are short oligonucleotide which are ligated at the end of sequence, which is used for sample recognition. That is why we are also calling this adapter as the unique identifiers for our sample. Okay. Then once you have like did the adapter ligation step, then you are going to send this fragments to the sequencing. Okay. Where you will get what? The raw data in the form of reads.
Hello, excuse me.
What are
Adapter
reads?
is hello, excuse me. Adapter is a primer, or like then what would be the difference between the primer and adapter here? Is it different oligonucleotides adapter here?
Yes, sir. Adapters and the primers are different, sir. Not the same.
Okay.
But the
So
role is same.
you meant to say like we have n number of fragments, correct? Then how do we can generate n number of adapters? Like so, can you please little bit explain more on adapter? what's the purpose for the adapter, how many adapters we need to generate. So could you please explain?
Yeah. Just wait. Can you first tell me what is the primer, sir?
Primer is nothing but like for PCR amplification, they used to have the primer. Like based upon that, the corresponding DNA fragments will be attached, and from that they will multiply. So forward primer, reverse primer, they used to do. It is
Yes,
basically
sir.
for PCR amplification.
Yeah. So, sir, these primers are again short oligonucleotide, which is very specific for a particular sequence, right? But
Yes,
what
yes.
is the main difference between the adapter and the primer is that adapters are also the short oligonucleotide, but they are universally labeled and universal in nature, but they are platform specific, sir. That is the difference between the primer and the adapter.
Universal adapter means like, do we have any maximum count, or like do we need to design like TNTP? They have D adapters. Sorry, A adapter, T adapter, C adapter, like this, or like it is having some sequence length like 50 sequence. What would be the size of the adapter, and how many adapter are like it is
That
specific?
the size thing part we will discuss later on, sir. Okay.
Okay,
Yeah,
okay.
in this session only. But the difference I would suggest, like these are adapters are the platform specific and the primers are specific for a particular sequence. So this is the main difference, sir. Okay.
Here, platform specific, do we have any meaning here? Like what we are meant?
Platform means, sir, like the sequencing platform the which you are running, like Illumina, for example, the Roche, if you are running, so that they are having a specific adapters for that. Okay. So that only you can run on that particular machine.
Or do we need to design adapter like any region kind of stuff that Illumina or like Roche they will give
No,
used to make?
sir, like you already getting the automatic kit. You are not designing that adapter, sir.
Okay, okay.
Yeah. So that part, like already, like the people who are very expert in the sequencing, that they guys are doing, not us. And automatically, like when you are running the sequencing, also you will get the primers with you. So these are not this only one primer you will get. You will get the hi-fi mix. So hi-fi mix is the like bundle of the primers there. Okay, the enzymes there. So automatically the kit, it is already the design. It is already designed for that particular machine. So we guys are not doing that part.
Okay, so the machine will take care all the things. Only
Yes,
thing
sir. Yes.
we need to put the sample. At the end of the sequencing, we will be getting the entire sequence.
Yes, sir. Yes, sir.
Okay.
If, for example, you are running on the Illumina or the maybe the Iron Torrent platform. So, for example, you are running the Iron Torrent platform. So they are giving you the hi-fi mix also, and also the adapters also. They will provide you. We are not designing that. But the cost of the sequencing is high actually.
You mean to say for NGS, if you use adapter, the cost of the sequence is high?
Yeah.
Okay,
If
okay.
you want to do starting from the isolation till the data analysis, the cost will be high, sir. So that is why sometimes people can't afford that. So they are just sending their sample to the sequencing.
Okay, got it.
Yeah. Now, yeah. So once you have sent to the sequencing, you will get this particular data, raw data, in the form of FASTQ file. So that we will discuss like in the pipeline. What is the FASTQ format? So raw data you will get in this format, FASTQ format. Okay. Then after that, the third step is the data analysis that the bioinformatics people do. So this part, the library preparation till the sequencing, it is doing in the wet lab techniques. Okay. So did you got the idea of this library preparation, sequencing, and the data analysis? But it depend on the aim, okay, of the application. So it will vary according to that application. So this NGS application also basically divided into DNA and the RNA. And in the DNA, you are having the DNA sequencing, metagenomic also you can study. If you want to study the gene expression, that you can do here in the RNA, that covered in the reference based, de novo based, right? So let's talk about the DNA-seq data analysis, where we are going to determine the order of nucleotide in DNA in the individual genetic code which we call it as the DNA-seq or DNA sequencing. So if you want to know the ATGC pattern in the DNA, we need to do the sequencing. But the aim of the DNA sequences is not just to know the DNA sequence, but also you can determine the variants in isolated DNA of an organism. But how we are going to identify the variants? With the help of the reference genome. So once you have this raw data, what we are doing, it is in the FASTQ file, right? Which is containing what the read sequence. Now we want to find out the variation in the DNA. So you already know that, for example, that particular data has been taken from human. Okay. This raw data. So this FASTQ file is from human. And for this particular data, we are going to find out what? The variants, right? So what we are going to do, we will download the reference genome of the human from the databases such as the NCBI, UCSC. So you already know how the bases are arranged in the reference genome, right? Of the human. So with the help of some tools, maybe like there are some mapping tools like HISAT2, BWA, Boitai. So these are some mapping tools. What it will do. So for example, this reference genome we have downloaded from the UCSC and it is of human. And they are having adenine here, thymine, and guanine. So with the help of this mapping tools, we are going to align this particular data on this reference genome. So it will align like this. So after doing the sequencing and after doing the alignment, the reads will mapped on the reference genome. And what happened? For example, on this first position, we already know there is a adenine on this reference genome, right? And after doing the alignment, what we saw on this particular position, all the reads are having what? The adenine. So From looking at this data, we can say there is no variation. But at second position on this reference genome, there is a thymine, and once we mapped these reads on the second position, what we saw, the reads were having what? The guanine. So tool is going to detect this particular thing. Okay, this guanine. So by looking at this position, we can say the thymine is getting replaced with the guanine, which we call it as what? The variant. So this is just a basic idea of this DNA-seq. And the tools, the detailed information we will discuss when we will download the data from the NCBI SRA. But for now, did you understand like what we are doing with this raw data? So there are so many things we have to consider, like that whether the reads have aligned properly or not on that particular position, whether that base is called properly by the sequencing machine or not. So all these conditions we also need to check, because if the base is not called or sequenced properly during the sequencing, so the analysis will be wrong. Okay. So that is why all these conditions we are going to check and identify the variants by aligning the reads to the reference genome, and we are going to perform the DNA sequencing, or we call it as the variant calling analysis.
Hello, excuse
So
me.
this, yeah, yeah,
Hello,
yeah.
excuse me. Can you write some sequence, like example 5 to 10 sequence, and like you have drawn in the MS
Mhm.
Paint, like so I couldn't visualize. So if you don't mind, like in Word itself, like our Notepad, you can write one sequence, like and another sequence you can write by below. And can you explain whichever you explained here? It will be really helpful for me, because here I couldn't visualize the, like you told variant mutation. Could you please explain once again? Like you put some sequences examples, and you can say like T is changed into, and do we need to consider what and all sequences it get changed, or like we need to see only one sequence it will get sequenced? Like why are we having multiple sequences? Like we are having first sequence read, correct? So
Yeah.
this read is like a set of sequences, or like what is the length of this read, or like it's a particular complete genome of a particular gene, like what does it read means?
No,
And
no, sir. See, like for now, I'm not discussing like in today's session. This is just a basic idea, the brief description I'm giving you. But later on, the thing which you are asking me, like we will discuss during the data analysis part. Okay.
okay, okay,
Yeah, this all things will be covered. Okay, the data analysis. So this is just a basic idea, like the T is getting changed with the guanine. That is the variation, because some of the people like they do not know what is the basic idea of this DNA-seq, right? So for them, like I'm just giving just a simple example, because otherwise like it will be a very huge task for them also, right? So the things which you are telling me, like how, what is the position of that reads, and how the analysis will be look, that part we will do when you will have or the results in your system also. Okay, then only you can correlate your data.
okay, got it.
Okay, we will discuss, sir. Don't worry, this is just a like first session on DNS, like the basic introduction, that's it. Automatically, well, like when you will have the results in your system also, you will have a idea, because we are providing the cutoff also, the filter also, because we cannot take all the reads of for the good quality, because some of the reads won't be pathogenic, right? We need to neglect all the benign ones. So benign is what, the when you see benign word, so it is not causing any disease to the patient. But if you see the pathogenic, so that is what, like the mutation which is happening in that particular patient, it is causing the disease for that. But So for this, we have to take some treatments immediately, not for this data, benign one. And we will discuss the, might be you have heard about the missense, synonyms. So that all part will be covered, sir, during the analysis, not here. Okay. Okay, so this variant analysis is divided into two parts. First is the whole exome sequencing. Second is whole genome sequencing. So any idea the basic difference between whole exome and the genome? Anything? What is this whole exome sequencing?
Exome stands for the part of DNA that is actually transcribed, the genes that actually express.
Yeah,
And genome
right.
includes introns also.
Yeah. So genome, as you see genome word, so this is the whole set of genes, and exome is what the whole set of exon, which is, as she said, the coding part, which is 1% of the genome. So 99% of genome is non-coding. So if you are interested to find out the variant in the coding part, that time you can go with this whole exome sequencing. So, sequence of all exons in a genome, we call it as the whole exome sequencing, and which is used to identify possible disease causing mutation. And if you want to find out the variation in different part of the genome, that time you can go with this particular application, that is the whole genome sequencing. So let's discuss the pipeline which you will going to perform in your system. So first step, we will see the data downloading, which will be in the FASTQ format. And that we are going to download from the NCBI SRA database. Then we will download the reference genome, which will be in the FASTA format. And that you can also download from NCBI, UCSC, Ensemble. So it's up to you which platform you want to use for the reference genome, you can use. Then once you have the data downloading, like data, you need to check what the quality, right? Whether your raw data is good or the bad. So that you are going to check. That is the quality control with the help of one tool, that is the fast QC tool. Okay. So with this fast QC tool, you are going to check what, whether your raw data is good or bad. If it is good, then it is fine. But what if it is bad? What is bad means here?
Has wrong results,
No, 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 no.
wrong sequence.
No. Bad means when you are having adapters or maybe the primer. So you are not interested to study about the adapter or the primer sequence, right? We are only interested to know about our interested sample, right? So wherever you will see the adapter and the primer, these are the unwanted sequence. This is like which you have used for only for the amplification part, right? So we are not going to study that particular things. So that is what the unwanted things that we are going to trim with the help of tool, that is the cut adapt. or maybe you can use the fast p. So there are a lot of tools, trimometric, trim galore. So it's up to you. Like if you are able to use this cut adapt, you can use cut adapt. The role of the tool will be same only. Okay. Then again, after removing the adapters and the primer from your data, you need to check again the quality, right? Then you will get a good quality. So once you have a good quality of your data, you are going to align the reads on the reference genome with the help of one tool, that is we are going to use the BWA. So once you have aligned that reads on the reference genome, then further we are going to perform the variant calling analysis with the help of GATK tool. So with this GATK tool, we are going to see what are the variants there. Okay, what is the change of the base in that particular sequence that we are going to see here in the GATK. But it is not possible like each and every data that is present in your raw data, it is causing what? The disease, right? Because there are thousands of, like there are multiple genes there. So each and every gene, it is not causing a disease. So that you can also filter the variants with the help of one tool, that is SNP SIF tool we are going to use. Then after you filter the variants, you need to know the annotation of the variants, right? What is the annotation? Any idea? In this variants, you will like you will come to know about the change of base T into C, right? But you want, if you want to know what is the gene that is present in that particular variants, or maybe the gene position, or maybe on the chromosome location, or maybe the biological functional pathway, all these network analysis. So all these things, we, if you want to know, that you can do with the help of this annotation while using this web tool. Okay.
Ma'am, can you repeat this annotation part?
Annotation, for example, you are having on your reference genome, that is the A, right? But in your mutant, like, or maybe in your sample, there is a change of base, right? And for this particular thing, you want to know what is the gene present here, right? What is the chromosome location? So all the annotation, the detailed information you want to know, that you can retrieve from using this web, whether it is present on exons or not. Okay. So that we will see the detailed information here in the 7th step while using the web tool. Got it? So these, like in coming 10 days, we are going to see these things in detail. Got it? The basic idea, did you got it? What we are going to perform?
Yes, yes.
Okay. So so everyone is having the Linux platform or maybe the WSL in their system. Do you have this?
No, ma'am.
Who's this?
Akanksha.
What about others? Lena?
Yes, ma'am,
Ratu?
I have.
Saima ma'am, do you have this WSL platform? Sarvanan sir, do you have this?
Yes, I do have Linux PC itself.
Okay, okay. Kritika?
Yes, ma'am.
Okay, so only Akansha is not having this. What about Abhishek sir? Okay. So meanwhile, whoever is having the Linux platform, they need to install the some tools using repository method. So you can install BWA. Second is SAM tools. Third is Picard tools you can install. So with the help of this command, you have to install sudo app hyphen get install and the tool name. Got it? So I will tell you how to install one tool. Sudo apt hyphen get install BWA. Enter. Give the password. Then it will be installed. Then just type a tool name and the manual will be opened. So these are the commands, or you can see the help file to run the BWA tool. Likewise, you have to install this Picard tools, SAM tools in your system. Can you try, all of you? Akansha ma'am, just remind me tomorrow. We will connect before the session and we will install this WSL in your also system because we require this Linux platform for this.
Okay, ma'am.
Okay, yeah. Meanwhile, others can just install this tool. If you will face any problem while installing this tool, just let me. Are you installing this tools, these three tools in your system? Just let me know once it is done. Okay, okay. Okay, then we can end the session here, and so make sure these tools are installed, and whoever is just not having the Linux platform, just ping me tomorrow. We are going to install like in your system also. Yeah, yeah, Rakhi, I will upload. Okay. Okay then. Bye bye.
Thank you, ma'am.
Yeah. So in the MacBook, you don't require Abhishek. So for that, there are different commands we will discuss during the session, maybe tomorrow or maybe like after that. Okay.
Okay, ma'am. Okay, ma'am. Thank you, ma'am. Thank
Okay.
you. Bye bye.
Okay. Bye.